Why do women live longer than men? Scientists at Lobachevsky University have come close to unraveling the difference in life expectancy between men and women. How profound are the differences between men and women? A question that has tormented the minds of scientists, poets and philosophers for thousands of years. Why do we react so differently to stresses, to changes in the environment, perceive information differently, and of course, why do women on average live longer than men? Regardless of race, place of residence, average life expectancy in the region, level of medical care, maternal mortality and other factors, always and everywhere women on average live longer than men. Somewhere this difference is 2 to 3 years, for example in Europe, and in some regions of Russia it reaches 8 years and even 12 years. Life expectancy may increase significantly, as in Japan, as a result of the development of medical technology and increased quality of life, but there is still a statistically significant difference between men and women. What is the reason? Scientists emphasize that the risk of age-related diseases depends directly on the age of the person, but not so much on the chronological, the one in the passport, as on the biological, the one that actually reflects the processes of accelerated or delayed aging. However, how to know one's biological age is still an open question. One of the most promising ways is the epigenetic clock. It turns out that the work of genes is regulated by chemical modifications of DNA. A gene can become blocked as a result of methylation of certain sections of DNA, and conversely, become active if these tags are removed. Amazingly, the methylation level of some genes increases or decreases evenly with age, bringing to mind the myth of the ancient Greek Moira, the inexorable weaver of the threads of fate. Measuring the level of methylation of certain DNA points, we can thus spy the internal age of the body's cells, find out how much their epigenetic clock shows. Is it possible that the epigenetic clock runs differently in men and women? A study recently published in the journal Aging analyzed age-related changes in DNA methylation in men and women. Such a study requires absolutely not trivial solutions for processing large-dimensional data, because each methylome analysis of human DNA methylation sites includes about 450,000 points, notes the study leader, Doctor of Physical and Mathematical Sciences, Pro-Rector for Scientific Work of the National Research University Mikhail Ivanchenko. It is impossible to solve such a problem without using complex mathematical methods of analysis. Researchers from Nizhny Novgorod State University showed that there are DNA sites, CpG sites, with different trajectories of age-related methylation changes in men and women. Analysis of such sites and the functional activity of the genes to which they belong indicates that these are not only loci associated with hormonal regulation, but also genes with gender already obtained cannot be overestimated. It is not only a pioneering study of gender-specific DNA regulation in the context of aging, but also a fundamentally new approach that reveals previously unknown molecular mechanisms of gender differences in longevity, the study of which in the future will help to prevent the development of age-related diseases.